Hi dear students, hope you are all doing great. So in this class, now we are going to learn about smooth muscles. We have already learned about skeletal muscles, we have already learned about cardiac muscles, and now it's time to learn the smooth muscles. How do you proceed in this class? Basically, what are the things that we are going to learn in this class? So we are going to learn the functional organization as we have learned in case of cardiac muscle in a physiological point of view, and then we are going to classify the smooth muscles initially. Then we are going to see the mechanism of contraction of smooth muscles as we have learned in case of cardiac and skeletal muscles. So let's proceed. The first question that logically would come to your mind is why this is called smooth muscle. Smooth muscle എന്നാണ് പറയുന്നത് അല്ലേ? It's called smooth muscle. Why? Because as the name suggests, it looks so smooth. That means it doesn't have any striations. So the name itself has something to do with the structure of the muscle that we are talking about. It doesn't have striations. You don't see any striations. So come, let's learn about the types of smooth muscles and how are they organized. Okay. So basically, there are two types of smooth muscles. What are the types of smooth muscles? We have single unit smooth muscle and we have multi-unit smooth muscle. So these are the different types of arrangement of the smooth muscles. As I said, when we come to learn about the detailed structure of a cell, we will learn that they don't have striations and they are very smooth. So this is something that you can see when you're asked about a smooth muscles. What you can write is this particular shape of a cell. See, they're spindle shaped. So every smooth muscle would be spindle shaped. If you take a single cell, they would be spindle shaped and there would be a nucleus. This is the nucleus of the cell. And this is the cut section of an artery. രക്തധമനി എന്ന് പറയുന്ന സാധനം നമ്മൾ കട്ട് ചെയ്ത് കഴിഞ്ഞാൽ അതിൽ കാണുന്ന മസിൽസ്. See, suppose we take an arteriole, we'll be seeing a lot of muscles like this. The inner layer would be made of muscles, and this would be the endothelial layer, and this would be the muscle layer. You can see. ഇങ്ങനെ പല പല സ്ഥലങ്ങളിലായിട്ട് നിങ്ങൾക്ക് സ്മൂത്ത് മസിൽസ് കാണും. ഫോർ എക്സാമ്പിൾ, നമുക്ക് ഏതൊക്കെ സ്ഥലങ്ങളാണ് പറയാൻ കഴിയും? ഒന്ന് നമ്മുടെ യൂറിനറി ബ്ലാഡർ, വി ക്യാൻ സീ ദ സെയിം ടൈപ്പ് ഓഫ് മസിൽസ്. ആൻഡ് ആസ് ഐ ഹാവ് ഡിസ്ക്രൈബ്ഡ് ഹിയർ, ആർട്ടറീസ്, സ്മോൾ ആർട്ടറീസ് അല്ലെ ആർട്ടീരിയോൾസിലെല്ലാം വി ക്യാൻ സീ ദിസ് ടൈപ്പ് ഓഫ് മസിൽസ്. പിന്നെ എവിടെയൊക്കെ കാണാൻ കഴിയും? നിങ്ങളുടെ ഗട്ടിൽ കാണാൻ കഴിയും. നിങ്ങളുടെ ഇൻറ്റസ്റ്റൈൻ, നിങ്ങളുടെ സ്റ്റൊമക്കിൻ്റെ വാൾസ്, നിങ്ങളുടെ ഇൻറ്റസ്റ്റൈൻ്റെ വാൾസ്, ലാർജ് ഇൻറ്റസ്റ്റൈൻ, സ്മോൾ ഇൻറ്റസ്റ്റൈൻ്റെ എല്ലാം ഭിത്തികൾ, ദ വാൾസ് ഓഫ് ദ സ്മോൾ ഇൻറ്റസ്റ്റൈൻ ആൻഡ് ദ ലാർജ് ഇൻറ്റസ്റ്റൈൻ, സ്റ്റൊമക്ക്, എല്ലാം നമ്മുടെ ഗട്ട് എന്ന് പറയുന്ന സാധനത്തിലെല്ലാം നമുക്കത് കാണാൻ കഴിയും. പിന്നെ നിങ്ങളെ ബ്രോങ്കിയോൾസ്, നിങ്ങളെ ശ്വാസനാളം എന്ന് പറയുന്ന ബ്രോങ്കിയോൾസ്, ഈ ബ്രോങ്കിയോൾസിൻ്റെ ഉള്ളിലും നിങ്ങൾക്ക് കാണാം. അപ്പോൾ ഈ വെൻ യു സ്റ്റാർട്ട് ലേണിംഗ് അബൌട്ട് സ്മൂത്ത് മസിൽസ്, 2-3 കാര്യങ്ങൾ ദീസ് ആർ വെരി ഇമ്പോർട്ടൻറ്റ് ദാറ്റ് യു ഷുഡ് നെവർ ഫോർഗെറ്റ്. ഒന്ന്, ദേ ഡോണ്ട് ഹാവ് സ്ട്രൈഷൻസ്. മറ്റൊന്ന്, ദേ ആർ ഇൻവോളണ്ടറി. അതാണ് വേറൊരു ഇമ്പോർട്ടൻറ്റ് പോയിന്റ്. ഇൻവോളണ്ടറി എന്ന് പറഞ്ഞാൽ നിങ്ങളുടെ ബ്രെയിനിൻ്റെ കോർട്ടക്സിൻ്റെ കൺട്രോളിലല്ല ഇതെല്ലാം. നിങ്ങൾ വിചാരിച്ചു കഴിഞ്ഞാൽ അനക്കാൻ കഴിയില്ല എന്നർത്ഥം. ദേ ആർ ഇൻവോളണ്ടറി. ബിക്കോസ് ഓഫ് ദാറ്റ്, what I mean by involuntary would be, in a physiological point of view നോക്കിക്കഴിഞ്ഞാൽ, that means that your cortex is not involved in the mobility of these muscles, അല്ലെങ്കിൽ അതിൻ്റെ ഫങ്ഷനിങ്. your cortex has no role, the cerebral cortex has no role എന്നാണ് അതിൻ്റെ അർത്ഥം. സബ് കോർട്ടിക്കൽ ആയിട്ടുള്ള സ്ട്രക്ചേഴ്സ് ഓഫ് ദ ബ്രെയിൻ, കോർട്ടക്സിന് താഴെയുള്ള, കോർട്ടക്സ് എന്ന് പറഞ്ഞാൽ ചെറിയൊരു പുറന്തോട് മാത്രമാണ് ബ്രെയിനിൻ്റെ. ഇപ്പോൾ കോർട്ടക്സിൻ്റെ താഴെയുള്ള സബ് കോർട്ടിക്കലായിട്ടുള്ള ന്യൂക്ലിയകൾ, ബ്രെയിൻ സ്റ്റെം, മെഡുല ഒബ്ലിംഗേറ്റ, അങ്ങനെയുള്ള പല പല സെൻറ്റേഴ്സാണ് ഇതിൻ്റെ ഫങ്ഷനെ നിയന്ത്രിക്കുന്നത്, അല്ലെങ്കിൽ അതിന് മുകളിലുള്ള പല പല സെൻറ്റേഴ്സ്. വി ലേൺ ഓൾ ദീസ് in your central nervous system. അപ്പോൾ അതാണ് ഈ സ്മൂത്ത് മസിൽസ് നിയന്ത്രിക്കുന്ന ന്യൂറൽ കൺട്രോൾ. ഒരുപാട് കൺട്രോൾസ് ഉണ്ട്. ഇൻവോളണ്ടറി ആണെങ്കിലും ഒരുപാട് കൺട്രോൾസിലെ ന്യൂറൽ കൺട്രോൾ എപ്പോഴും would be by the autonomic nervous system. എന്താണ് പറയുക? ഓട്ടോണോമിക് നെർവസ് സിസ്റ്റം, ANS. ഈ ഓട്ടോണോമിസിറ്റി ഉള്ള നെർവസ് സിസ്റ്റം ആണ് കൺട്രോൾ ചെയ്യുന്നത്. ആൻഡ് ദാറ്റ് ഈസ് മേക്സ് ദ സ്മൂത്ത് മസിൽസ് ഇൻവോളണ്ടറി, ദാറ്റ് ഈസ് നോട്ട് ഇൻ യുവർ കൺട്രോൾ. ഇത്രയും ഓർത്തിരിക്കണം. ഈ രണ്ട് കാര്യങ്ങൾ എപ്പോഴും ഓർത്തിരിക്കണം. Now we'll learn about single unit smooth muscle and multi-unit smooth muscles. How are they organized? What is the difference between single unit smooth muscle and multi-unit smooth muscle? ഈ സിംഗിൾ യൂണിറ്റ് സ്മൂത്ത് മസിലിൻ്റെ ഏറ്റവും വലിയ പ്രത്യേകത എന്ന് പറഞ്ഞാൽ, I'll just draw and show you how a single unit smooth muscle will look like. Smooth muscles will be arranged in spindle forms like this. അട്ടിയായിട്ട് ഒരു ഗ്രൂപ്പായിട്ട് നമുക്ക് ഇങ്ങനെ കാണാം. This is a group of single unit smooth muscle. ഒരു 3D drawing ആണ്. That means cells ഇതിൻ്റെ അടിയിലേക്ക് പോകുന്നുണ്ട്. Just packed together. This ഒരു single unit ആണ്. I'm drawing here another single unit. They are also packed in a spindle fashion. Another point. എന്താണ് സിംഗിൾ യൂണിറ്റിൻ്റെ പ്രത്യേകത? We have a cell body here. I'm going to draw for you a neuron. I have drawn a neuron. This is the neuron. This is the cell body of the neuron. The axon is coming. And this is going to get into these smooth muscles. These axonal endings. These are axon endings. These are going to run through the smooth muscles. അപ്പോൾ ഇവിടെ ഓർക്കേണ്ട കാര്യം, and in this unit, in another unit, what you can find is another nerve cell is going to supply this. For a single unit, this is one unit. For this one unit, a single nerve with different axons. അപ്പോൾ ഇതൊരു ഒറ്റ nerve axon ആണ് ഇതിലേക്ക് വരുന്നത്. And for another second unit, unit 1, unit 2, this is another single unit smooth, and that has a separate second neuron. This is how the neural supply is organized. And mind you, this is an autonomic neural supply. When you compare it to skeletal muscle, that was a cortical supply. It was coming from the cerebral cortex. But in this case, these smooth muscles are not supplied by the cerebral cortex. അപ്പോൾ അതിന് എക്സാമ്പിൾ എന്തൊക്കെയാണ്? ദിസ് ടൈപ്പ് ഓഫ് ഓർഗനൈസേഷൻ്റെ എക്സാമ്പിൾ എന്തൊക്കെയാണ്? എക്സാമ്പിൾ യൂട്രസ്, സ്റ്റൊമക്ക്, അല്ലെങ്കിൽ സ്മോൾ ടെസ്റ്റൈൻ ആ ഭാഗം, ആൻഡ് യുവർ ബ്ലാഡർ. ഇതൊക്കെയാണ് കോമൺ എക്സാമ്പിൾസ് വേർ യു ക്യാൻ സി എ സിംഗിൾ സപ്ലൈ. Coming to a multi-unit smooth muscles, ഇതിൻ്റെ പ്രത്യേകത, they are made up of discrete, separate smooth muscle fibers. ഇവിടെ നിങ്ങൾ സ്മൂത്ത് മസിൽസ് എടുത്തു കഴിഞ്ഞാൽ, they are all present as a group. They are all discretely present together, but the importance is they don't have gap junctions. They don't share the informations. അതുകൊണ്ട് തന്നെ they all get nerve supply from different neurons. ഇപ്പോൾ ഒരു ന്യൂറോൺ തന്നെ, what does it mean is that here you might have a neuron which is supplying the axon which runs towards this. Same time, another neuron can also run to this. ഒരു ആക്സോൺ ഇതുപോലെ ബ്രാഞ്ച് ചെയ്ത് ഇതിലേക്ക് തന്നെ സപ്ലൈ ചെയ്യാം. അതുകൊണ്ട് തന്നെ ഒരു കോംപ്ലിക്കേറ്റഡ് ആയിട്ടുള്ള ഒരു ഓർഗനൈസേഷനാണ് മൾട്ടി യൂണിറ്റ് സ്മൂത്ത് മസിൽ ഉള്ളത്. ഇതാണ് ബേസിക് ഡിഫറൻസ് ബിറ്റ്വീൻ എ സിംഗിൾ യൂണിറ്റ് സ്മൂത്ത് മസിൽ ആൻഡ് മൾട്ടി യൂണിറ്റ് സ്മൂത്ത് മസിൽ. മറ്റൊരു വ്യത്യാസം ഈ ഗ്യാപ്പ് ജംഗ്ഷൻസിൻ്റെയാണ്. ഈ സിംഗിൾ യൂണിറ്റ് സ്മൂത്ത് മസിലിൻ്റെ പ്രത്യേകത, ഒരു യൂണിറ്റിലേക്ക് you get a nerve supply, that means it will spread along the whole unit because of presence of gap junctions, because these muscle cells here would be arranged in close association, and there are gap junctions between the cells, they are connected. But in case of multi-unit smooth muscles, there are no gap junction bridges, they are separately present together in a group as separate cells, they don't have communication between each other. അതിനുള്ള എക്സാമ്പിൾസ് ഏതൊക്കെയാണെന്ന് ചോദിച്ചാൽ, we have the iris, ciliary body of the eye, the muscles of the larger airways, the precapillary sphincters, pyloractus muscle. ഇതെല്ലാം എവിടെയാണെന്നൊക്കെ നമ്മൾ ഭാവിയിൽ പഠിക്കും. ഇപ്പോൾ ഓർത്ത് വെച്ചാൽ മതി, കുറച്ചൊക്കെ നിങ്ങൾക്ക് മനസ്സിലായിട്ടുണ്ടാവും. You will learn about all this precapillary sphincter. I don't know whether you know about all this precapillary sphincter, pyroerector muscle, ഇതൊക്കെ എന്താണ്, ciliary body of the eye, എല്ലാം നിങ്ങൾ വിഷൻ പഠിക്കുമ്പം you will learn about all this. So it's beyond the scope of this class to discuss each in detail. You can also divide these smooth muscles based on the pattern of contraction. ഈ പാറ്റേൺ ഓഫ് കൺട്രാക്ഷൻ വെച്ച് നമുക്ക് പറയാൻ കഴിയുന്ന രണ്ട് തരം സ്മൂത്ത് മസിൽസ് ആണ്. വൺ ഗ്രൂപ്പ് ഓഫ് സ്മൂത്ത് മസിൽസ് വിച്ച് റിക്വയർസ് എ ടോണിക് കൺട്രാക്ഷൻ. നമ്മുടെ ബോഡിയിൽ ചില സ്മൂത്ത് എപ്പോഴും ഒരു ചെറിയ തോതിൽ കൺട്രാക്ട് ചെയ്തിരിക്കണം. ദർ ആർ സം സ്മൂത്ത് മസിൽസ് വിച്ച് ആർ റിക്വയർഡ് ഇൻ ദ ബോഡി, വിച്ച് ആർ ഓൾവേസ് റിക്വയർഡ് ടു ബി ഇൻ എ സസ്റ്റെയിൻഡ് സ്റ്റേറ്റ് ഓഫ് പാർഷ്യൽ കൺട്രാക്ഷൻ. Always required to be in a sustained state of partial contraction. For example, blood vessels. Blood vessels. The blood vessel muscles and the arterioles, okay, they should be always partially contracted. Otherwise, you the tone of the vessel. BP എല്ലാം ഫോളോ ചെയ്യാൻ കാരണമാവും. അപ്പോൾ എപ്പോഴും ടോണിക്കലി കൺട്രാക്റ്റഡ് ആയിരിക്കണം. ഇത് നിങ്ങൾ ജനിച്ചു കഴിഞ്ഞാൽ മരിക്കുന്നവരെ എപ്പോഴും കൺട്രാക്റ്റഡ് ആയിരിക്കും എന്നർത്ഥം. ഇറ്റ് മീൻസ് by the time you are born till the time you are dead, it will be always in a state of partially contracted state. On the other hand, we have phasic contraction muscles. Idu namak arterioles okay ulla smooth muscles, the smooth muscles of the arterioles an example aanu. Going to phasic contractions on contract rhythmically and intermittently. You need some part of the body, the smooth muscles in some part of the body to be having a rhythmic contraction. ഒരു rhythmic contraction എവിടെ ആവശ്യമുള്ളതെന്ന് ചോദിച്ചാൽ നിങ്ങളുടെ ഗട്ട്, നിങ്ങളുടെ ഇൻറ്റസ്റ്റൈനിൽ ആവശ്യമുണ്ട്. ഇൻറ്റസ്റ്റൈൻ എന്തിനാവശ്യം? നിങ്ങളുടെ ഫീക്കൽ മാറ്റർ എല്ലാം പുറത്തേക്ക് പോകണമെങ്കിൽ, ഡൈജഷൻ പ്രൊസീഡ് ചെയ്യണമെങ്കിൽ ഫ്രം സ്റ്റൊമക്ക് ഫുഡ് ഹാസ് ടു ഗോ. രാവിലെ നീക്കുമ്പോൾ കറക്റ്റായിട്ട് പോകണമെങ്കിൽ ഇതിങ്ങനെ മെല്ലെ മെല്ലെ മൂവ് ചെയ്യണം. It should move slowly from your stomach, should move towards the small intestine, then to the large intestine, to the rectum, and finally it has to go out. അപ്പോൾ അതൊരു rhythmic movement ആണ്. അതിനെ നമ്മൾ പെരിസ്റ്റാൾട്ടിക് മൂവ്മെൻറ്റ്സ് എന്നൊക്കെ പറയും. We learn, I hope you have already learned in your GIT about all these things. അപ്പോൾ ഇത് നടക്കുന്നത് ഈ rhythmic ആക്ടിവിറ്റി ഉള്ളതുകൊണ്ടാണ്, ഇൻ്റർമിറ്റൻറ്റ് rhythmic ആക്ടിവിറ്റി. അപ്പോൾ അതിന് എക്സാമ്പിളാണ് നമ്മുടെ ഗട്ട് ആൻഡ് ഇൻറ്റസ്റ്റൈനൽ സ്മൂത്ത് മസിൽസ്. അപ്പോൾ ഈ രീതിയിൽ നമുക്ക് ക്യാൻ ഒബ്സർവ് സ്മൂത്ത് മസിൽസ് ആസ് ടോണിക് ആൻഡ് ഫേസിക് ഗ്രൂപ്പ് ഓഫ് സ്മൂത്ത് മസിൽസ്. ടു എ very subtle level we can make classification as single unit smooth muscles and multi-unit smooth muscle. Another form of a grosser level, at a very grosser level, we can again divide them into different forms of arrangements. For example, you can see only some smooth muscles are only arranged in a circular fashion. Some parts, bronchioles ഒക്കെ എടുത്തു കഴിഞ്ഞാൽ സർക്കുലർ ആയിട്ട് മാത്രം അറേഞ്ച്മെന്റ് ഉണ്ടാവും. സർക്കുലർ അറേഞ്ച്മെന്റ്. ഇപ്പോൾ സർക്കുലർ അറേഞ്ച്മെന്റ് എങ്ങനെ ഉണ്ടാവും? ഇറ്റ്സ് ലൈക്ക് ദിസ്. ദിസ് ഈസ് യുവർ വിൻഡ് പൈപ്പ്. സപ്പോസ് ദിസ് ഈസ് യുവർ വിൻഡ് പൈപ്പ് എന്ന് വിചാരിക്കുക. എയർ ഈസ് ഗോയിങ് ഇൻ. എയർ ഈസ് ഗോയിങ് ഇൻ ത്രൂ ദിസ്. സോ ഇവിടെയുള്ള സ്മൂത്ത് മസിൽസ് എല്ലാം അറേഞ്ച് ചെയ്യുന്നത് ഇൻ എ സർക്കുലർ ഫാഷൻ ലൈക്ക് ദിസ്. ഈ വാൾസിൽ ഇങ്ങനെ അറേഞ്ച് ചെയ്തിട്ടുണ്ടാവും. Smooth muscles will be arranged in this way. അപ്പോൾ ഇങ്ങനെ അറേഞ്ച് ചെയ്യുമ്പം when they contract, what happens is this will happen. They will become narrow. Contraction will make them narrow. അതാണ് circular smooth muscles arrangement കൊണ്ട് these type of arrangements would be seen in those areas of the body where you require, where a hollow ഒരു passage requires narrowing. അങ്ങനെയാണെങ്കിൽ you require circular arrangement. You might also find smooth muscles which are arranged both circularly and longitudinally in the same place. Suppose the same cylinder which I have drawn is your gut. നിങ്ങളുടെ ഗട്ടാണെങ്കിൽ ഗട്ടിൽ നിങ്ങളുടെ ഇൻറ്റസ്റ്റൈൻ ആണെങ്കിൽ there will be both circular arrangement as well as longitudinal arrangement. Smooth muscles would be arranged in this way. ഇങ്ങനെ അറേഞ്ച് ചെയ്തിട്ടുണ്ടാവും. അതുകൊണ്ടുള്ള ഗുണം എന്താ? So when these longitudinal muscles and circular muscles contract, the same would become like this. It will also narrow and it will also shorten length. Narrowing and shortening. Both this occur. We can also find places where the same smooth muscles are arranged in circular manner, longitudinal manner and in oblique manner. Perfect example would be that of the urinary bladder. Urinary bladder, which is a bladder, would be like this, right? ഇങ്ങനെയുള്ള bladder ആണ്. പുറത്തേക്ക് urine will be collected here. We need to void it out to go out. ഇങ്ങനത്തെ സിറ്റുവേഷനിൽ what will you find? When a contraction occurs, what will you find? And see, it become like this, contracts. So we have circular arrangement, longitudinal arrangement and oblique arrangement. All this will make this thing smaller like this. ഇതുപോലൊരു സ്ഫെറിക്കൽ ആയിട്ടുള്ള ഒരു ബ്ലാഡറിനെ നമുക്ക് ചെറുതാക്കണമെങ്കിൽ ഒരു 3-dimensional സാധനമാണ്. ഇറ്റ്സ് എ സ്ഫിയർ, ഒരു ബോൾ പോലത്തെ സാധനമാണ്. So we also have longitudinal arrangements. This is oblique arrangement, this would be your circular arrangement and this would be your longitudinal arrangement. അപ്പോൾ ഇങ്ങനെ 3 രീതിയിലും അറേഞ്ച് ചെയ്ത് കാണാം. Just to give you a conceptual idea regarding all these things. Coming to the real sense, smooth muscle, this is a single cell. Now you are going little bit into the cell and trying to understand why was it called smooth, or ഈ നോൺ സ്ട്രൈറ്റഡ് അപ്പിയറൻസിൻ്റെ ഒക്കെ റീസൺ ആണ് നമ്മൾ പഠിക്കാൻ പോകുന്നത്. വി ആർ ഗോയിങ് ടു ലേൺ ഹൗ ദിസ് ഇസ് ബേസിക്കലി അറേഞ്ച്ഡ്. സോ വാട്ട് ഈസ് ഷോൺ ഹിയർ ആർ 2 സ്മൂത്ത് മസിൽ സെൽസ്. അവിടെ കാണിച്ചിരിക്കുന്നത് ഒന്ന് അതിൻ്റെ സെൽ മെംബ്രെയിൻ. ദേ ഹാവ് സെൽ മെംബ്രെയിൻ ആൻഡ് ദേ ഹാവ് ഷോൺ ദ മയോസിൻ ഫിലമെൻറ്റ്സ്. ദിസ് ഏരിയ, ഈ ഏരിയ നിങ്ങൾ ശ്രദ്ധിച്ചാൽ നിങ്ങൾക്ക് മനസ്സിലാവും. ഇവിടെ 2 ബ്ലൂ ഡോട്ട്സ് കാണാം, 2 blue dots. What are these blue dots? They are called dense bodies. Here also we have dense bodies. The dense bodies, they are also made up of proteins. The actinin എന്നൊക്കെ പറഞ്ഞിട്ടുള്ള ആൽഫ ആക്ടിനിൻ ഇങ്ങനെയുള്ള സാധനങ്ങൾ കൊണ്ട്, ഫോർ എക്സാമ്പിൾ ഇതൊരു ജസ്റ്റ് ടോൾഡ് യു എൻ എക്സാമ്പിൾ, പഠിക്കേണ്ട ആവശ്യമൊന്നുമില്ല. ഇങ്ങനെയുള്ള പ്രോട്ടീൻസ് കൊണ്ട് ഉണ്ടാക്കിയ ഒരു ഉണ്ടയാണത്. പ്രോട്ടീനാണ്, ബേസിക്കലി പ്രോട്ടീൻസ്. ഈ ഡെൻസ് ബോഡീസ് a very important component of smooth muscles which you don't find in skeletal muscle or cardiac muscle. And actually to these dense bodies you have connection of contractile proteins. Your actin filaments are connected to these dense bodies. You have your myosin filaments which forms the center part between the 2 dense bodies, and this is at this place where your real contractions occur. അതുപോലെ ഒരുപാട് പോയിന്റ്സ് ഉണ്ട്. We can see lot of dense bodies in a single muscle, smooth muscle cell. And here you will also find your nucleus. ഇങ്ങനെയാണ് ഇത് അറേഞ്ച് ചെയ്തിട്ടുള്ളത്. If you see very closely the dense bodies, you will find ഈ ഡെൻസ് ബോഡീസ് കാണാം. യു ക്യാൻ സീ ദ ആക്റ്റിൻ മയോസിൻ ഫിലമെൻറ്റ്സ്. ഇവിടെയാണ് നിങ്ങളുടെ കൺട്രാക്ഷൻ നടക്കുന്നത്. ഇറ്റ്സ് എ പ്ലേസ് വെയർ യു ക്യാൻ സീ യുവർ കൺട്രാക്ഷൻസ് occurring. Point. So all points contract at the same time. That means the whole smooth muscles after contraction would look like this. Is contracted, right? Is contracted as a whole. That was regarding the structure. And in the cell membrane, you will also find a very interesting thing. When you compare this with your T tubules in skeletal muscles and your cardiac muscles, what you find here are small invagination here, which are called calveolae. And you will also find sarcoplasmic reticulums inside. See, all these things are called sarcoplasmic reticulums. And you can also find, for your interesting thing is that they are not well developed. Not well developed. അതാണ് അത് വേറൊരു പ്രത്യേകത. These are the two other important points that you should remember when you start learning smooth. Take a reason എന്താണെന്നുള്ളത് we come to it. Coming to the neuromuscular junction in smooth muscles, they are different. എന്താണ് പ്രത്യേകത എന്ന് ചോദിച്ചാൽ, these are the important things that you should. A typical neuromuscular junction is not present. So what is happening? So you already know they are innervated by autonomic nerve system, and these nerve fibers branch diffusely. I've shown in my previous slides, a single neuron branches, the axon branches diffusely, and they don't end on a smooth muscle. Smooth muscle directly poi end ചെയ്യുന്നില്ല. Cases they don't end. They don't have an end feet അല്ലെങ്കിൽ ഒരു motor end plate. They don't have all these things that you commonly find in skeletal muscles. But instead, what would you find are small rounded structures. They are called varicosities. See here, this is an axon which is going from a cell body. The axon in ഒരു ബ്രാഞ്ചിൽ find small varicosities. ഈ ഉണ്ട കാണുന്നില്ലേ? These are called varicosities. And all these varicosities would have your synaptic vesicles. You have your synaptic vesicles, and this will rupture down and release its neurotransmitter, which are either acetylcholine or norepinephrine. I've learned about all these things. They will be released. So that is what happens, or this is how a smooth muscle is different in its nerve supply. So varicosities basically, and they are releasing their neurotransmitter. And such an arrangement is called synapse en passant. ഈ വേർഡ് എവിടെയും കേൾക്കുകയാണെങ്കിൽ don't get confused. പേടിച്ചോടൊന്നും വേണ്ട. synapse en passant എന്ന് ആരെങ്കിലും പേടിപ്പിക്കാൻ ചോദിച്ചു കഴിഞ്ഞാൽ, synapse en passant എന്ന് പറഞ്ഞാൽ ഈ ഒരു passing on of a single axonite with varicosities. അതാണ്. So from a synapse on the surface of smooth muscle cell pass on to make similar contacts with other cells. A neuron എങ്ങനെയാണ്, how a neuron makes contact with other cells അല്ലെങ്കിൽ adjacent smooth muscle cells. ഈ ഒരു particular arrangement ആണ് what we call synapse en passant. And you also will find a different pattern of arrangement of the autonomic nerve system in multi-unit smooth muscles. What would you find in multi-unit smooth muscles? Find varicosities, but these varicosities are separated from muscles cell membrane by 20 to 30 nanometers. This would be similar to the neuromuscular junction of skeletal muscles. Bring faster contraction in multi-unit smooth muscle. So faster contractions in multi-unit smooth muscle is possible because of this one-to-one arrangement of varicosities. ഓരോ സെല്ലിലും വേറെ വേറെ ആയിട്ട് ചെല്ലുന്നുണ്ട്. That is the speciality of or the importance of contact junctions in multi-unit smooth muscles. Now we'll move on to the electrical properties of smooth muscles, which would not be an elaborate topic, as we have learned in cardiac muscle and smooth muscle. We have learned these electrical events in very detail. Here it's not learned in that detail. We have already learned, and most of the mechanisms remain unclear. So we won't go to the details of the electrical properties of smooth muscles. But always remember, the resting membrane potential remains between -50 to -60 millivolts. And the most important thing to remember is that the smooth muscles show all the different potentials that we have learned. One was the spike potential that you usually find in the nerve and skeletal muscle. In smooth muscles, you will find action potential with a plateau type, which was usually seen in cardiac muscles. And you'll also find slow wave potentials. Slow wave potentials which are almost similar to those which were found in pacemakers like SA node, AV node. We had found these pre-potentials and pacemaker potentials. All these would be very similar to the slow wave potentials that are seen in some forms of smooth muscles. So basically we find all the three forms of potentials in smooth muscles. So coming to the first potential, which is a spike potential, you will see this in urinary smooth muscles. Very smooth muscles. The amplitude will be very low. The duration would be 10 to 50 milliseconds. And regarding spike potential, this is very important that not only your neuronal signals can produce spike potentials. Not only new nerve signals, you can have your blood vessels releasing a hormone. So suppose this is your urinary smooth muscle cell, you would have an axon coming like this with varicosities. This might be moving like this, a neuron with varicosities. You can also show that a blood vessel is passing nearby, and the blood vessel releasing is releasing a hormone. A hormone H can be released, and this would have receptors over the smooth muscles, and they also can make the smooth muscles develop a spike potential. You can have stretching of the muscles. See, the same muscle, if you stretch like this, so in case of urinary smooth muscles, stretching would be caused only by one thing, and that is your urine. Now, when urine fills in your urinary bladder, the bladder smooth muscles will stretch, and simply by this stretching, the stretch can develop an action potential on the smooth muscle. If you take an electrical stimulator and just stimulate your bladder, this can also develop a spike potential. So these are the different faculties which can develop a spike potential in a smooth muscle. Coming to the next form, which is the plateau potential, which is seen in smooth muscles. And which smooth muscle you will see it? You will see it in uterus. Uterus, you can see a plateau potential. Plateau potential, that means uterus, there will be a potential which lasts longer. A potential which lasts longer. And there will be prolonged contraction. A repolarization does not occur immediately. Why I am not stressing on this? We have already learned the potential changes occur in a plateau phase. All the electrical events we have already learned in cardiac muscle. Same electrical events will occur in a plateau potential smooth muscle also, for example in uterus. So that was your second type. And then we have the slow wave potentials. The third one being the slow wave potentials. These are seen in your visceral smooth. Viscera എന്ന് പറഞ്ഞാൽ ഇങ്ങനെ വയറിൻ്റെ ഉള്ളിലുള്ള സാധനങ്ങൾ. So basically what we are mentioning here is the muscles of the gut. Muscles of the gut എന്നാൽ നിങ്ങൾ intestinal wall, stomach wall ഇതൊക്കെയാണ് ഉദ്ദേശിക്കുന്നത്. And low wave potentialsന്റെ പ്രത്യേകത, they are almost like your the pre-potential and your SA nodal potential. നമ്മളവിടെ SA നോഡിലും AV നോഡിലും കണ്ട ഓട്ടോമാറ്റിക്സിറ്റി കാണിക്കുന്ന തരത്തിലുള്ള പൊട്ടൻഷ്യൽസ് ആണ് you will find in intestine, because what you require in intestine is spontaneous rhythmic contractions. What you find is that the membrane potential itself will have these spontaneous rhythmic fluctuations, and that is going to give the smooth muscles a rhythmical contraction. So this will appear in regular intervals. The contractions are going to come by itself at regular intervals and they are going to move forward. So why this happens? So why this membrane potential is having a rhythmic fluctuation is due to the waxing and waning of pumping of positive ions like sodium. Positive ions in the gates would be open and closed, and that is the reason for developing a rhythmic fluctuant type of membrane potential. And what you can find is these small waves that you find initially, these waves cannot form a contraction. These are electrical events. They are not the contractions. Electrical aanu. And when they reach a threshold level, they cause the spike elevation of membrane potential, and that also will occur in a rhythmic fashion, and this will induce contraction of the smooth. So when the peak of the slow wave potential rises in positive direction from -60 to -35, there your action potential develops and spreads over the muscle mass, and this is going to cause the contraction. That was the third type of potential which develops. The action potential appears rhythmically at the rate of 1 to 2 spikes at the peak of each slow wave, cause rhythmic contraction of smooth, and these are called pacemaker waves. ഇവിടെ നമുക്ക് same usage കാണാം, pacemaker, like that we saw in SA node. You can it's very easy to learn this if you compare it with the activities, the electrical activities that occur in the pacemaker cell of the heart. അതുപോലൊരു pacemaker activity ആണ് ഇവിടെ intestinalum കാണുന്നത്. Yes. Coming to the stretch activation of stretch induced contraction of muscle, of smooth muscles. എങ്ങനെയാണ് ഉണ്ടാവുന്നത്? Why this happens? That's the next point we are discussing. Sufficient stretching of the skeletal smooth muscles can cause a spontaneous action potential to develop, and that can cause contraction of the smooth. Why this happens? Because whenever there is a stretch, there are stretch induced channels. Smooth. Take this muscle over the smooth muscles, You have specialized channels. They are called stress channels. SCN എന്നൊക്കെ പറഞ്ഞ് കാണാം, stretch channels. When stretch occurs, these channels are activated and they will allow calcium to get into the. അതുപോലെ തന്നെ we have another channels we have learned about, ligand gated channels. We have learned about voltage gated channels. അല്ലേ, അങ്ങനെ പലതരം സ്റ്റിമുലസ് കൊണ്ട് ഓപ്പൺ ചെയ്യുന്ന ചാനൽസ്. Voltage can induce opening of channels. Ligand can induce opening of channels. In the same way, stretch can induce opening of. അങ്ങനെയുള്ള സ്ട്രെച്ച് ഇൻഡ്യൂസ്ഡ് ചാനൽസിൻ്റെ പ്രസൻസ് ആണ് കോസിങ് contraction of smooth muscles just by stretch. Coming to the next point, which is called the junctional potential. The junctional potential. What is junctional potential? Let us learn what is junctional potential. So multi-unit smooth muscles ആണ് ഇത്തരത്തിലുള്ള junctional potential, like muscles of iris, pilorector, as we already discussed. The nerve stimulus will release neurotransmitters and they call depolarization of the and there will be contraction.
11.
No action potential. No required. The action potential is not required for contraction. That is the point that is relevant here. The fibers are too small to generate action potential. നെർവ് പഠിച്ച സമയത്ത് നമ്മൾ പറഞ്ഞിട്ടുണ്ടായിരുന്നു ഇതുപോലൊരു കൺസെപ്റ്റ്, എക്സൈറ്റേറ്ററി പോസ്റ്റ് സിനാപ്റ്റിക് പൊട്ടൻഷ്യൽസ് എന്നുവെച്ചാൽ ദീസ് ആർ സബ് ത്രെഷോൾഡ് സ്റ്റിമുലസ് കൊടുക്കുമ്പോൾ നെർവിൽ ഉണ്ടാകുന്ന ചേഞ്ചസ് ആർ നോട്ട് സഫിഷ്യൻറ്റ് ടു പ്രൊഡ്യൂസ് ആക്ഷൻ പൊട്ടൻഷ്യൽ. ഇങ്ങനെയൊരു ചേഞ്ചസിനെ പറ്റി നമ്മൾ പറഞ്ഞിട്ടുണ്ടാവുന്നു. ഇവിടെ നമ്മൾ പറയുന്നത് എ ലിറ്റിൽ ബിറ്റ് ഡിഫറെൻ്റ് ആണ്. ഇവിടെ പറയുന്നത് ആക്ഷൻ പൊട്ടൻഷ്യൽ ഡെവലപ്പ് ചെയ്യാതെ തന്നെ, വിത്തൗട്ട് എൻ ആക്ഷൻ പൊട്ടൻഷ്യൽ, എ സബ് ത്രെഷോൾഡ് ലൈക്ക് സ്റ്റിമുലസ്, എ നെർവ് സ്റ്റിമുലസ്, മേ ബി ഐദർ സബ് ത്രെഷോൾഡ് or threshold, need not produce an action potential. The small potentials, they are called junction potentials, can actually cause contraction. അതിനെയാണ് നമ്മൾ junctional potentials പറയുന്നത്. And they are capable of depolarizing the muscles and causing contraction. Coming to the excitation contraction coupling that you see in smooth, it's a little bit different and very important to learn. And it's different from smooth muscles, different from your cardiac muscles and your skeletal muscles. എന്താണ് ആ ഡിഫറൻസ് എന്നാണ് നമ്മൾ പഠിക്കാൻ പോകുന്നത്. ഇത് പഠിക്കുന്ന കോണ്ടെക്സ്റ്റ് എന്താണെന്ന് ചോദിച്ചാൽ, this is the context. Smooth muscles, as I've already told you, that there are tonic contracting smooth muscles. That means for a very long period of time they are in a state of partially contracted. They are in a partially contracted state for a very long period of time. അപ്പോൾ അത്രയും പീരീഡ് ഓഫ് ടൈമിൽ കൺട്രാക്ട് ചെയ്തുകൊണ്ടിരിക്കണമെങ്കിൽ അത്രയും എനർജി അവിടെ പോവില്ലേ? വെരി ഒബ്വിയസ് ഡൗട്ട് that should come into your mind. We will lose a lot of energy as ATP, അല്ലേ? It has to be in contracted state for a long period of time. ില്ലേ എനർജി ലോസ് വരില്ലേ, ബോഡിക്ക് ഭയങ്കര ബുദ്ധിമുട്ട് വരില്ലേ എന്നുള്ള സംശയങ്ങളുണ്ട്. അപ്പോൾ അത് എങ്ങനെയാണ് ഈ ലോ എനർജി സ്റ്റേറ്റിൽ നിന്ന് ഹൗ ഈസ് ദിസ് മസിൽ ഏബിൾ ടു കൺട്രാക്ട് ഫോർ എ ലോങ് പീരിയഡ് ഓഫ് ടൈം എന്നുള്ള ഒരു പോയിന്റ് മനസ്സിലാക്കാനാണ് വൈ വി ആർ ലേണിംഗ് excitation contraction coupling in a possible for a smooth muscle to contract for a long period of time without consuming much energy. That's the whole point of മനസ്സിലാക്കേണ്ട കാര്യം, the contraction coupling is very slow as per the slides. The slow process ആണ്. Calcium, very important contraction. Already learned this in cardiac muscle too, that extracellular calcium plays a very important role. And I have already told you that there is an underdeveloped sarcoplasmic reticulum that you find in smooth. Underdeveloped. So that means Actually, the smooth muscles completely rely on the calcium. So this calcium has to get in and do the excitation to the contraction. This calcium, so a stimulus should come in that should cause calcium get in and then contraction should happen. Stimulus, calcium getting in and contraction should happen, and then has to relax. This is how it goes. Now we are going to learn about the excitation, contraction, coupling, and the steps. So first thing that you learn should be about the stimulus, and we have already learned about this. If you want to learn about stimulus more, I would have to explain this, that one part of this would be the neurotransmitters. The neurotransmitters. Second one would be the hormone. Idellam stimulus. What kind of stimulus can you have? You can have nerve signals, cause neurotransmitter release. You can have hormones directly. You can have your extra electrical stimulation. Electrical stimulation can happen. You can also have your stretch. ഇതെല്ലാം can cause calcium. So ഇതൊക്കെ നമ്മുടെ സ്റ്റിമുലസസ് ആണ്. എല്ലാതരം സ്റ്റിമുലസസും can cause all the excitation, contraction. ഇത് കഴിഞ്ഞാൽ നെക്സ്റ്റ് നമ്മൾ വരുന്നത് കാൽഷ്യമാണ്. Okay, after stimulus, then we have to discuss about the calcium that comes in. The calcium അവിടെ നമ്മൾ പറയേണ്ടത് കാൽഷ്യം എന്താണെന്നുള്ളതാണ്. The source of calcium ions has cause contraction, mainly the calcium, the EC, gated, the ligand gated calcium channels in the cell membrane. All will come into play here. There is an involvement of the intracellular stores, especially in the sarcoplasmic reticulum. But since this is not developed properly, the amount of calcium that sarcoplasmic reticulum will be very less. So this has a very minimal role when compared to the extracellular calcium. Yes. So then what happens? So let's think in terms of a hormone or a neurotransmitter that comes to a receptor and how the action will proceed. See, we can see here also we can see a new hormone neurotransmitter acting. Here also we can see a hormone neurotransmitter acting. But what is this? What is this hormone neurotransmitter? We can call them as a first messenger. We will learn about all these first messenger and second messenger systems. So this forms just an introduction for you about these messenger mechanisms. This is the first messenger that brings informations from the brain through the neural system or from the subcortical structures as hormones. They now enter and come to the surface of the smooth muscle. So this is your basically a smooth muscle cell, and on the surface you have them hitting on the receptor. See the receptor here. Now this receptor can be linked. What is this? Oh, this is just a protein. This is called a G protein. So this protein is linked directly to a ligand gated calcium channel. So while you stimulate your G protein, the receptor hormone will form a complex. This hormone receptor complex or the receptor complex can directly activate the G protein, and this can open the calcium channels, ligand gated calcium channels, and calcium can come in. One route by which calcium can come in. Same hormones or neurotransmitters have receptors which acts through the same G protein but in a different way. they can also activate certain second messengers. These are the messengers that go into the cell and will convey the message from the surface of the cell to the inside of the cell, maybe at this spot, maybe at this spot. So from here, the message, the letter has to go and reach here. Message should reach the sarcoplasmic reticulum. These are those chemicals, the PIP2 converting into IP3. We have a lot of such second messenger systems. We will learn about these second messengers in detail when you learn your endocrinology. Now just understand this IP3 is a second messenger, and this second messenger is going to activate the store operated calcium channels. These are calcium stores inside the sarcoplasmic reticulum, and so these channels will be called the store operated calcium channels. So these are open and calcium comes out of stores. This is how calcium comes in from the sarcoplasmic reticulum out into the cytosol of the. We have also the voltage gated calcium channels. If you give a direct stimulation, electrical stimulation, this can activate the voltage gated calcium channels, and this way also calcium can come in. So it all, we have different ways where calcium can enter, and depending on the stimulus that and the place where the smooth muscle is located, the mechanisms vary, and the cells also will arrange itself in such a way as to receive the appropriate stimulus, and this is how calcium gets in. Now since now the calcium has got into the cell, and now the calcium is in the cytosol, it's time for the muscles to contract. The force of contraction of smooth will highly depend on the extracellular calcium, as already discussed that this very very low probability in case of smooth cell. Everywhere calcium has to come from outside to inside, extracellular. So ECF calcium forms a very important part. Now after this, how is that the muscle contract? The mechanism is little different from those mechanisms that you find in cardiac muscle and your skeletal muscle. So this is how it happens. The calcium that comes in is going to release the calcium stores through the store operated calcium channels, or the calcium which is stored is also going to dislodge from inside the sarcoplasmic reticulum into the cell cytosol. Now this calcium is going to bind immediately to a binding protein called calmodulin. Calcium is, if it is free inside the cell, then it will be very difficult for the cell to handle the calcium. The calcium always binds to different proteins, and calmodulin is that protein, calcium binding protein that you see inside all the smooth muscles, which will initiate the contractions. So this complex is going to activate your myosin light chain kinase. That was your myosin light chain kinase. Now what happens to your myosin light chain kinase? Now the myosin light chain kinase transformed. Now this is activated, and this is inactive myosin light chain kinase. So now the myosin light chain kinase is activated. And what does this myosin light chain kinase do? The myosin light chain kinase is going to act. What is this I'm drawing? What is this I'm drawing? And act. Here is your actin, and here is your myosin. Basically here myosin. Okay, so what does this guy do? Myosin light chain kinase എന്താ ചെയ്യാൻ പോണത്? Myosin light chain kinase go into phosphorylate your myosin. This myosin that you find here is actually not like this. It's actually made up of lot of proteins, light chains, heavy chains, lot of chains. Just a bunch of proteins. Just a bunch of proteins. അതിൽ ഒരു protein, one protein is myosin light chain. So myosin light chain phosphorylate. What happens is the phosphate, a high energy phosphate gets attached, and this myosin light chain kinase does this job. And where does this high energy phosphate come from? It comes from ATP. So what does the myosin light chain kinase do? Converts ATP to ADP. That's your ADP. So this phosphate and attaching itself to myosin. The phosphate is attaching to myosin. At this point, you should terribly note this fact that in skeletal muscle, you have a separate area for ATP to bind to myosin. You have a separate area for the ATP to bind to myosin. But here, we have a separate kinase. Smooth muscles, we have a kinase, a protein which actually breaks down ATP and takes out the phosphate, and the phosphate is planted on myosin light chain. And now when this phosphate comes, this suddenly gets energy, very happy now, it gets the energy, and now very smart, and it attaches actin, and it can slide over, like this, can slide. Now that makes contraction, well and good. What about the relaxation? The relaxation is happening by another protein, is called myosin light chain phosphatase enzyme. Let's go to the slides and revise contraction and come to relaxation now, or become confusion. Okay, let's go to the slides. Binding of the acetylcholine to muscarinic receptors. Calcium influx into the cell. Calcium binds to calmodulin complex. Activates calmodulin dependent myosin light chain kinase. Catalyzes. This is going to cause the phosphorylation of myosin light chain kinase, already described as phosphorylation, and binding of actin, myosin to actin. Actin slides of myosin. That was the contraction. Now let's come to relaxation. Relaxation, what is happening? Dephosphorylation of myosin light chain kinase. Dephosphorylation brought about by myosin phosphatase. So how does this happen? Come to the same diagram here. Yes. So here we already have the phosphates attached, and now what is required is we have to remove this phosphate, and to remove this phosphate we have the enzyme called myosin phosphatase, that is phosphatase. So myosin phosphatase myosin phosphatase, the one is going to remove this phosphate, and then that means we have relaxation. We have relaxation. Just think, try to take out your memory and think in skeletal muscle, how was this happening? If you remember, in skeletal muscle, there was no myosin phosphatase. There was no myosin phosphatase. The relaxation and further attachment was occurring when a fresh ATP was getting attached to the myosin head. I hope you remember this. A fresh ATP coming and attaching to the myosin head, that is going to block or cut connection between actin and myosin. And but then when the ATP breaks down and it gets its high energy phosphate, again with use this phosphate, it again makes reaction. This was what was happening in skeletal muscle. Here, for the relaxation to occur, we just need myosin phosphatase enzyme. If you have very high concentrations of myosin phosphatase enzyme, that means there is faster relaxation. Suppose myosin phosphatase enzyme is very low, kept very low inside the cell, that means the cell, the smooth muscle cell, remain in the contracted state for a very long period of time until the cell gets enough myosin phosphatase. I hope this concept is clear. So the myosin light chain kinase dephosphorylation and relaxation. For this part to occur, you need myosin phosphatase, and the concentration of myosin phosphatase is a very factor for regulating the tone of your smooth. Coming to the latch bridge mechanism. So what I have explained just now is latch bridge mechanism. Once contracted, the muscle can sustain contraction for much less energy and uses 1 by 300 the energy of skeletal muscle, and sustained tension low energy. അപ്പോൾ ഇവിടെ ഈ എനർജിയുടെ കാര്യം മനസ്സിലായെന്ന് I hope you have understood what I mean by this energy usage. You just compare the skeletal muscle movement with the smooth muscle movement. You can easily make out that ATP molecules, when a fresh ATP molecule attaches to the binding site on the myosin, relaxation occurs. And when the ATP hydrolyzes to ADP and phosphate, the myosin can then utilize the phosphate and make a contraction, proceed with the contraction. For the relaxation to occur again, a new ATP attaches. At that point, suddenly it relaxes. It gives the phosphate, contracts. Here, during the relaxation period, there is no involvement of a new ATP being coming because it's completely followed up by your myosin phosphate that dephosphorylates the myosin light chain. It's actually myosin light chain phosphatase, myosin light chain phosphatase or myosin phosphatase. That's called the latch bridge mechanism. That almost finishes your smooth muscles, and let us conclude by also discussing the length tension relationship in smooth muscle. So we will take the example of urinary smooth muscles to explain the length tension relationship. So if a unitary smooth muscle is stretched It first exerts increased tension. The stretch is maintained, the tension gradually decreases. That's the thing that we are going to explain now. Now we are going to see how this happens, and we are taking the example of a urinary smooth, and we are going to learn this special feature of smooth muscle called plasticity of smooth muscles. Basically, that's nothing but adaptability of smooth muscles to a new length. That means basically the smooth muscles has particular property of expanding and maintaining the state, maintaining its length. That's what we are going to learn and understand length tension relationship. This is seen in hollow visceral organs like bladder, as I have already explained. Maintain the same amount of pressure inside the lumen with stretch changing. How will you explain this? We explain this like this. What will happen? Got a small balloon like this, and if you make the balloon big like this, not like that, it's under high tension like this. It would be at under high tension. Balloon would be almost like this, very big and about to burst like this. Or the borders would be in very tension, high tension, right? What is the pressure inside? It would be very high, right? Increased pressure, and this is a normal pressure. But this is not the case, in case of a urinary smooth muscle. So don't never compare a blowing of a balloon and the state of a fully contained balloon with a urinary bladder. Ever find anyone explaining a urinary bladder like a balloon? Don't take it to your brain, because that's wrong. Because in case of in case of a balloon, what happens inside is, as you blow, the walls are at high tension. What does it mean? If you just release, what will happen is it comes back very fast to its original length. It want to retain its length at a very fast rate. That's not the case with the urinary bladder. The inside pressure never be increased, but instead the smooth muscles would stretch by itself, and it would be remaining in a very happy state of stretching, so that the inside pressure is not allowed to rise very long period of time. That is what we are going to learn with the help of very simple graph. Okay, let me show this first, then we'll go to graph. A sudden increase in volume of urine in urinary bladder cause pressure. I think this would happen, right? There will be stretching of smooth muscles in the bladder, and the pressure returns to original level within a very short period of time, within seconds or within milliseconds, even with continued stretch. That is what we are explaining here in this diagram. You see this diagram, this is very obvious. This is called a cystometrogram. The gram kandalum, that is recording. All grams. So we have cystometrogram. In this cystometrogram have an x-axis and a y-axis, like all graphs have an x-axis and a y-axis. You can see here we have the volume, and here we have the intravesicular pressure. What is the pressure inside the bladder? Pressure inside the bladder, this P is here, and the amount of urine that is filling inside the bladder is here. See what happens. See, initially, when suddenly when the urine is getting filled, you can see the pressure also start increasing. The first phase, which is the 1A phase. But suddenly what you find after this phase is that after this, the pressure which we would normally seen in a balloon will go like this. Balloon graph would go like this. As the volume, the air increase, the pressure also increase. But here it's not like that. Here in our body, our smooth muscles behave in this way. The volume increases, the pressure having almost no change. And this about 100 ml. Until the bladder is having 400 ml, your pressure till 20 centimeters, way below 20, even around 10 point. But after that, at after 400, when this rises, now this becomes the limit, almost the limit of stretch of your muscle. And then what you see, very sharp pressure. At this point, you will feel inside your body that you should find a washroom. So it has to pass. So the, the feeling of passing urine comes this point. So what is learn about this detail when you learn your renal physiology. At this point, what is more important is a peculiar type of length tension relationship. So a length tension relationship is completely different from this cardiac muscle. Here, your bladder, you're finding a complete form of a relationship, length and tension. It's very, and that kind of behavior that you find here because of the peculiar stretchable nature of the muscle arrangement that you find bladder, that describes a length tension relationship. Not much increase, not much increase in intravesicular pressure until bladder is well filled up to when it is stretched, pressure is not going up. You have to write it, produced not to and is. We produce either same pressure is well maintained here. Find the pressure doesn't change. The pressure is maintained at that range of around 10 water till point of 100. That describes your length tension relationship, and that finishes your class on smooth. Thank you.